So Ellen, if I may start with you also, so it's all fresh what you have shown in our mind. What is missing in, so to say, in terms of interoperability, of making the connections between the different labs in Europe, in the States, so what shall we, I mean, what is the next step? What do we need to develop?
Well, different people have different focus on the research. If you're interested in clinical research, if you're interested in clinical research, then your primary search dimension is going to be patient. And you want to have all of that information that we've just been talking about to be referable to the patient. So you want these particular cell lines have to be allow us to interrogate this individual patient's cellular profile against those standards. But it has to be that patient, not a particular cell by itself. It's the patient cell profile. So that's from a clinical research perspective. If you have a more basic research direction, you want to understand the nature of cellular relationship between cellular profiles and, for instance, neuroreceptor and neurotransmitter profiles, then you're not so interested in the individual patient. It's a different question.
But would you see any obstacles? I mean, what are we missing? So we have now developed HIBALL. It will finish next year. There are other initiatives also developing, I mean, these links. eBrains is one, Tom. But what is not yet there? If you could dream about
this may be a contentious statement, but we're all guilty of not doing due diligence. Everybody reinvents the wheel in their own particular context to get funding, and they don't really want to hear that someone else has already done that. That's just the nature of the scientific mind. I think we're all guilty of this.
Yeah, maybe something I could add. I think what is also important, besides building a bridge and establishing tools and standards that work together, creating a setting where this will continue to be the case, right? Because infrastructure is changing, standards are changing constantly, and I think it's also important that we make sure that this goes with these modified environments in the future, right? So. Like if you have a software stack that makes sure we have an interoperability across the continents here, how can we make sure that this will also be the case when we set up the next supercomputer, right? That this will still be there then and not get lost on the way.
I have a question for you, Alan. I mean, this incredible world you're describing, how do you plan to connect it with the healthcare system? With the healthcare world, in fact, with the hospitals and all these places?
That's very difficult. Yeah, this is a very thorny question because of GDPR, any kind of regulatory oversight. It's from an external perspective, it looks like Europe has managed to paint itself into almost complete stasis with so many mutually contradictory regulatory oversights from different countries and different organizations. I don't know how we're going to get out of this. I think, Amy, I think you probably have a more direct experience than I do of this problem.
Of sharing data? Yeah, it can be tricky. Yeah, I mean, you know, I would love to make our data openly available without a data sharing agreement, but we are just absolutely not allowed. And like there are then issues with like human data versus animal data versus in vivo data, and yeah, we are constantly hitting battles of different like legal requirements versus research bodies and funders. Yeah.
It changes not only within country, it changes within jurisdiction within a country. In the Canadian context, Quebec is much more restrictive than Ontario about the dissemination of data, and they make little distinction, as far as I can see, between raw data and derived data.
Yeah.
Well, if you take this to its logical extreme, people trying to analyze, for instance, the UK Biobank would be required to get REB approval for every question that they ask, which is madness. This would almost shut down research. If every time you thought of a new hypothesis, you had to write an REB approval to process de-identified ones and zeros.
Yeah.
But this is the situation we're in, where the technology has outpaced the regulatory oversight. It's going to get worse when we come to internationalization of projects. Technologically, we can do it today. We can technically take data from anywhere in the world and put them into common repositories, and in theory get access to millions of individual data subjects, which would allow us scientifically to address some very, very large questions about data representation. Problem is political and oversight and regulation. All of those things which are intended to help are going to be a serious problem for open science.
I mean, so with the, you were talking about how you've got Biobank on. How's that going to be affected by their new regulations?
Well, it's the UK Biobank is a special case as we speak. For those who might not know, that the UK Biobank is a champion of open science over the last decade in making this huge resource available to the world. And lots of sites around the world have developed infrastructures to allow them to analyze those data in their own setting. As of a couple of months ago, the UK Biobank has declared that they will stop all of that. And to get access to the UK Biobank, you will have to go through a single source called DNA Nexus, which supports their research access portal, and all access to the UK Biobank will go through that. The stated reason for that is because of the concerns about data breaches and lack of security in sites out there. So they want to rein in the dissemination of the data, which have a much higher level of oversight and control. The consequence of this is it's going to be a lot more expensive for people out there to get access to and process UK Biobank data. This is a live issue. It's almost inevitable as you see the conflict between the principles of open science and data security. And the legal dimension. I don't know how we resolve this.
Perhaps Claire? Yeah.
Yeah, I think it's really interesting, the consortium that we're a part of, the Human Organ Atlas Hub, but also very linked in with the Human Cell Atlas program and things. And I think they're really good places to go to and to look at these kind of legal frameworks and started putting these things in place and working. But I think to come back to the very first question, I think one of the things that drives these kind of links and making stuff interoperable and shareable is collaborative projects between these spaces, and in particular, multimodal imaging is a great example of that, because by collecting certain data sets in one site, certain data sets in another site, and the entire project team needing to work on them, it forces these issues to be resolved in kind of practical ways that are actually useful. It doesn't, the legislation obviously then is a whole other layer on top of it, but I think collaborative projects are some of the best ways to actually move these things forward practically.
And Claire, when I see, your beautiful reconstruction and think about the vessel structure, I could imagine that many neurosurgeons are dreaming to get these vessel trees, because it goes much beyond angiograms that we know. So what, in your view, are the perspectives of using this ultra-high resolution down to the capillary level for patients for surgery, for example?
So I think we've definitely been doing a lot of work in the vasculature space. We've been focusing on the kidney initially with this modality, just because it's easier to image smaller data sets, and showing how you can extract these vascular networks with your CNN-based tools, and then you can do flow simulations. I think the biggest challenges for that kind of work is it's incredibly labor intensive, and I think it's a problem in general with vasculature. It's very easy to get a very nice looking vascular network that is beautifully visual for presentations. It's incredibly difficult to get a really properly well connected vasculature that you can meaningfully simulate blood flow or other things on. And it's an area that I think a lot more work is needed. We have done some segmentation, so the hippocampal arteries, we've just released a paper on recently, looking at those regions, and it's something that we're just building up enough training data for to be able to apply the methods.
So we have also, I mean, we know from the Allen Brain Institute the amazing genetic data. Mike, you worked there for quite a long time, and you have observed how, how the mouse data came together, now the human brain. So do you see that we are already there to predict human genome data based on existing mouse data and maybe sparse existing human brain data at the level of single cells? So predicting, I mean, regions that we could not yet analyze with such a detail because it's extremely expensive, for example, but then combining the different species data and sparse human data to do something else. So maybe experimental research is not needed in a few years. Can you comment?
Well, I don't know if I would go that far, but yeah, I think that, now the viability. There's a few issues here. I'd like to come back to other questions. I think that in a little bit, but let's talk about that first. But the viability of dealing with human tissue has greatly improved in recent years. We can now record in vivo from human cells. We have very strong ability. We can manipulate kind of genes in human tissue. We've learned a lot from mouse studies. There's good alignment in single cell data across species from mouse to marmoset, macaque, and human. This is a work in progress, and now we're looking at different systems in the human. But I think just to come back, so it is I don't think in any way model organism research is really replaced by this, but it is we get a better understanding of how to map between the species and understand what are differences in human from some other species. But I think I just want to come back a little bit to some of these earlier points about this interoperability issue, which I think is really fundamental. That the part of the problem is that different bodies establish themselves, get funding, and due to the nature of having to do things and to distinguish oneself, each group ends up we try to expand what we're doing to encompass putatively large parts of solutions of things. And a lot of times, not everyone can do everything, right? And so the question is, how do we open kind of these communication sort of infrastructure and avenues so that we can take advantage of things that really reflect really best on the thing. Take, for example, BigBrain, right, is that as a, the optimal kind of or highest level anatomic kind of, infrastructure that we have. We don't, now in this BICAN, the Brain Initiative Cell Atlas Network, we are using we're using this, kind of static human atlas that we had from Songlin Din to basically anatomically kind of select data from different regions. There's also a plan to use functionally driven parcellations from some of David Van Essen's work, as well as new work from Nancy Kanwisher and kind of active kind of living patients. But this is still it's fairly limited in its sense of ability to kind of pick data from these sources. And I think we don't address the structural issues which are done well with BigBrain and Hawrylycz and things like that. So I'd like to see and discuss ways that these kinds of initiatives can be aligned better. I think the interoperability to take advantage so that we're really driving toward these questions which are really insurmountably difficult.
Perhaps, Jeff, you would like, to comment on this as well?
Well, yeah, interoperability is a dream, but sometimes very difficult to achieve. But there's something that we've learned from the HBP story, which is that nowadays a lot of people have decided that data should stay in some places that are secure and software travel. But I think that some postmortem brains should travel across the different modalities to create some bridges. It's very important to link different kind of information. Otherwise we have incredible resolution for some modalities, but it's very difficult to recollect different brains. This is one of the tricks I think we discovered during HBP.
Well, I think that's really something that is really worth to think about. And I mean, we are partly starting this. Yeah? So brain is scanned at Neurospin. Then it is shipped to Amsterdam, and then it goes to Jülich. And I could imagine also having some exchange with Claire, yeah, so that we really are collaborating on this way, or also at the genetic level, because then we have the possibility also to overcome inter-subject variability, yeah, which is really a tricky question, not only at the level of folding pattern, but also at the cellular level. And the inter-subject variability at the cellular level is not so obvious in my view. I mean, cells seems like cells. And even the differences between neurons from mouse and macaque and human brains were taken a couple of years ago very, so to say, closely to each other. And then came out the first studies that show, the dendritic trees are different. There might be specific neurochemical characteristics. Matthew Larkham worked in that respect. And then the question came, what is really species-specific and what is not? So when we have a look to one and the same brain, bringing together our different methods, we have, of course, a possibility to answer these questions. And that means also, to be interoperable, because at the end we want to exchange the data sets at the end and bring them together. But these, I think, would be already two answers. Be interoperable. We cannot, so to say, try to systematically develop, in my view, a worldwide network where we all have supplementary jobs, and that probably will not work. But what we can do, we can be very transparent and clear where are the contact points, the links, and then, so to say, set up specific experiments to answer these questions. The homology question I find really interesting, and when you are going to the connectome level, do we have a possibility to understand in a more precise way and to quantify where are the similarities between cross-species comparison and where are the differences? So the question of homology is a question that is very clearly defined in biology, I would say, and here we have some bones that are developing and some other bones. That's rather, I would not say simple. For the brain, it's much more complex. So would you see that interoperability and experiments within one and the same species would help to answer this question?
Yeah, so cross-species comparisons, I think, at the connectome level, fascinating. There's so many similarities in certain modalities between species, but then at the whole brain level, it's so different. And I think Nicole has done some really interesting work recently in the hippocampus and trying to look across macaque and human and using data from BigBrain for that. And I think that, they're having the kind of cross-modal information can also be really interesting. And I think actually coming back to what you're talking about between subject differences, there's then obviously between cross-species differences. And even within the same modality, there's cross-group differences on, we can both acquire NISL data, but that data is often very different when we actually look at what the images show and do the tools work well between the different animals and between the different groups, and actually adapting the same tools across the different data sets and across the different species can be really difficult in itself, right? So really whether or not you're comparing like for like can be a challenge, I think.
So you can comment, but I would also now like to open it to the audience, and I would like to ask you to address your questions to one of the speakers and give your arguments, pros and cons of what you're listening to. So please don't be shy. Go ahead and be part of this discussion. Jeff, you wanted. No.
Katrin, one more just comment first regarding that. You know, I think that we now know pretty well how to align and map genetic data between species. We have pretty good models of how to identify structure and organization and how it homologously maps. And so there's an interesting question right there is how do we correspond that with what we know about anatomic and connectivity data. So there's really a very interesting area of study there, I think.
So Timo, you are, so to say, you're the person behind the Atlas technology, and you are developing now for many years tools and software to have a multi-level and multi-scale Atlas. What are, so to say, would it be interesting for you? Is it possible from a technical perspective to link this kind of atlas to the Japanese brain initiatives, to the Chinese brain initiatives? I mean, how to make these different atlases compatible with each other? Does it make sense? Is it possible? Or is, so to say, is it so much work that maybe that's not the best strategy, and to keep it rather, so to say, independent from each other? So as an atlas person, there are many atlases. What do you think about it?
I think we do need to connect it. I think it is very important to do that. And a little bit, I agree a little bit with Alan that it's always difficult to get people focusing enough on the compatibility as compared to doing your local grants and strategies. But it is very important to do it, and it's also very difficult to do it. I think that the INCF is a very notable institution in that regard, because they keep pushing us to do meetings on these topics and discuss these topics. There's another one coming up in this month in Texas, where this is actually of concern. And I think we have made great progress there, right? So we have developed metadata standards. We're developing on bridging the metadata standards, like we have developed open minds in eBrains, and we are interacting with people in the US and Canada, how these things are linked. But at the same time, it's difficult, especially if you come to really the anatomy and really the regions and so on. So you need to integrate many sources of knowledge there. You need to work on what is known about homologies, what are spatial overlaps of different parcellations, and how are they computed. There are so many levels to this problem that I think we'll have some work to do for the coming years there. Yeah. But the short question is, I definitely think it's very important to link these systems.
When I think about this question about interoperability, software, tools, infrastructure, wouldn't we also need a changing, I would say, culture in our scientific collaboration that supplements publications of good scientific papers with more visibility for data publications, with more visibility also for software publication? Well, it's a little bit, developing a software that is really well running and is robust and stable. You do not get so many merits at this moment, yeah? While when you have a nice science paper, a deep insight about the mechanism, that's rather easy. So do you see that we need to make it also, that also software development, tools development approaches would play a larger role, or is this a little bit naive to assume it because it does not sell good enough?
Goes to me the question again? Yeah. So I would really love that this becomes more prominent, and I think we can see that in all the contributions here. Also, what Alan is saying about the tools in Canada, I think it would be great. And for data, I think it is a bit more established now, but still not. We're not yet fully there, right? So in many cases, we see that people have produced a very good and useful piece of software where actually there's a lot of scientific expertise going into how that is designed, just as you often do it in designing the protocols for your data acquisitions. But very often, people still wait with the publication until they actually have themselves also done the study with it to publish it at a sufficiently high impact level. And I think that's it would be good to disentangle that so that you really could reach the same level of impact with a software only. I think things are moving there, but we're not yet at the right level.
I think as well, just to add, like I think the ongoing support is a really hard thing, right? Like there's one thing of producing data or software and then shoving it out to the world, but ongoing support of training people and providing, help and feedback and keeping the software up to date, like that takes a huge amount of resource and often isn't very well, praised by publications, right? You can't write a publication that we supported more people. Hopefully they then do exciting research and that's how it comes out. But I think this is, I've been amazed by the number of resource and people here supporting all of this amazing work. But yeah, I think that's something that really needs more recognition.
Yeah, and I wanted to come back on the point about the data. I think it's interesting that you're thinking that the data recognition is a bit more advanced than the software. It's funny, from the way I see it, the software is almost a bit more advanced in recognition than the data, which just shows the viewpoints. But yeah, I think definitely like data curation, the manual process of data, like a lot of data curation in QC, can be a very manual, very time-consuming process. And I think within the scientific community, it's like rendered as a technical, non-scientific piece of work, and a phenomenal amount of it is incredibly difficult to do, incredibly scientifically difficult to do, unless you do that, and then we're all really keen to release our data sets worldwide. You release rubbish data sets worldwide that have poor quality, and then you can massively impact the quality of the science that comes out of it.
Well, we had this problem also in the Human Brain Project for quite some years, and the solution was that we invented some kind of high-level support teams, because indeed it's not only time-consuming, it's also not so clear from the beginning which type of data do you need to unambiguously define a data set. Yeah? What is really needed? And Lubert Zehl from our team now in Brussels, she has developed the Open Minds metadata scheme, and I think that's good progress in that direction, at least to having a framework to describe data in a meaningful way. But most of the researchers, or many of the researchers, are not aware that this is not only half an hour, but it's really a lot of work also to set up the experiment from the very beginning in such a way that you are able to do it. Would you like to comment also, Mike, or how you are solving it at your place? So did
well,
you
actually,
develop?
that was the point that I was going to make, is that science has changed dramatically in the last generation. Now it's industrial strength, and you need the kinds of teams approaches that the Alan has championed. I think most of us have struggled to put together the informatics team that will support the multimodal data integration and analytics. Most labs have 1.5 postdocs to do that. It's not enough, but the Alan has seen that a long time ago. So.
Well, we don't really have a perfect solution to that either, as it turns out. I think that at the Allen Institute, largely we put a lot into data generation. We put quite a bit into packaging data to put it out there in some sense. But a lot of the types of data that we do aren't necessarily, there's different challenges with large-scale imaging data, which require a lot more infrastructure and a lot more, techniques for visualization and for manipulation. We don't do that as well as some groups do. And we don't actually spend nearly as much on analysis as you might imagine. We actually have not really, compared to data generation, we don't spend as much on that. I think that it goes back to problems of where neuroinformatics lives in the grand scheme of brain science, and it's not really well appreciated from funding sources or even from scientific perception of its its value. I make this really joke that, that the definition of neuroinformatics is trying to aid in the understanding of the most complicated piece of matter in the known universe. But then I add on, everyone's heard that before, but I add on with a fraction of the resources that have been put into Microsoft PowerPoint. And there's a lot of truth in that, that in fact that we don't spend, data funders don't really, we regard as if we don't really believe in our data. If you believe in the data, then you would probably spend more time to really archively, really, ensure its viability, its interoperability. If you believed it had value, you would take care of it like that. Except what scientists tend to do is that it's almost as if we know from the outset that this is imperfect, and we just wait for the next new technology that can shine a little bit more light on something. So I think that we have to address that, right? And we have to understand that what is the value of the data that we have? How is it made operable? How can we share it? Not have each group trying to do everything, but have groups really kind of work on things that they're good at and to make it to interoperable with others. And I think that's a really, and then to your point, Katrin, you should identify smaller projects rather than try to solve the grand integration thing. Perhaps, with respect to the resources described here, let's say, and the BRAIN initiative, identify a smaller piece which could be mapped together and could address a problem. And I think if we have a project like that we could identify, that would set a certain amount in motion, that would be helpful.
I have a question maybe on a related but slightly different topic. And something I worry about with all these public data sets is data set decay. So if I download HTTP data and I run 20,000 statistical tests and I publish the one p-value that I get that's below 5%, then I'm the bad guy because I haven't done multiple comparisons. But if 20,000 people download HTTP independently and each run their tests, then that's fine. And I guess it's a really hard problem to solve because how are you going to keep track of what tests are being run on the data sets that we release? But at the same time, it does seem important because otherwise we risk just publishing a bunch of false positives without realizing it. So is that something that you guys worry about? Is something you think about? What solutions do you envisage? I know, for example, like with the UK Biobank, by requiring the registration of hypotheses, that's one way of mitigating that. But on the other hand, like Alan mentioned, that might slow down the rate of scientific discoveries. So, a question for you all.
I'm reminded of the discussion that we had yesterday about the role of AI, that AI potentially will be able to allow us to take multimodal data and find clusters that our puny brains would never have conceived of. Then the challenge is for us to retrofit a model onto those clusters that we found. But at least you can see how that might be an iterative way of hypothesis generation, model generation, and model confirmation in a way that takes us beyond what we can preconceive as a model interpretation. And such a strategy of big data cycling and iterative model development and more sophistication, it's going to get better when we can add in literally millions of individuals from around the world. So there is, I think, some confidence that the brain foundational model, which is being talked about but hasn't yet happened, that will allow us to get insight that we have hitherto been unable to do. That will be a consequence of aggregating very large amounts of data beyond the scope of any one lab, indeed any one country.
Thank you very much, Ellen. This was almost like a concluding work also, and I would like to now go around and ask everybody what your conclusions are, and perhaps you can link it to the question, what is the foundational model of the brain, so how it would look like, and why would you like to build it? So, Timo, just your last words. I mean,
yeah, with the foundational model, you already indicate that AI is doing a great. Is important there, and I really think that we see so many people now working on embeddings, and I think that has a great potential to bridge across the species and the scales, and also to maybe help us with gaps in the data. So I think for me an important direction is now to work on understanding well what these models are doing and make sure we, since that requires a lot of computation, to go in the right directions there in terms of how we build the AI models.
I think for the foundation model of the brain, I think one of the exciting things from my point of view, which, the data hungriness of such a model will force more data interoperability, more data sharing, and data curation, because to build these foundation models, really good data curation is a first process. And so I think even the attempts in making a foundation model, you end up with a much better homogenized interoperable set of data sets.
Yeah, it's a kind of a big question. I don't know, but I'm trying to imagine it. I'm not even fully understanding, I think, what we would mean by that. There's the anatomic aspect, there's systems kind of interaction, there's genetic interaction. How all this data would come together, I don't think we're so close to even kind of the right conceptualization of what such a model would be. I'm kind of more back in this idea of can we use AI toward really enhancing our understanding of data integration, and phrased in a somewhat more modest kind of goal. I think it's for me that's a little bit too big of a step on some sense from where we are, but I guess I'd like to see kind of more concrete demonstrations of how we can bring some of these multimodal data together into more a consistent model and, kind of have them be interoperable in that way.
Well, I guess I would be more confident in things that are looking like foundational model dedicated to specific questions where we have still some understanding of what we are trying to do, rather than pouring everything in something. Because for the language, it was something where there were some very simple pretext tasks to train these models, but for the brain, I'm afraid we don't have this kind of task nowadays.
Yeah, I think for me, what's really exciting is this idea of, creating these kind of digital phantoms and being able to think about, what are the parameters that we need to answer our question? And then based on the observations that we have, what is the probability of it being one thing versus another? And I think there's really exciting opportunities within clinical domains of, if I really care, is this consistent with inflammation versus, I don't know, some other kind of clinical outcome? And I think being able to build into these probabilistic models of, how does our digital phantom relate to the data that we can observe, and being able to build them from unique data sets that have tons of different modalities and then use that to predict in more restricted domains. I think that's what's exciting for me.
Thank you all very much for this discussion. I mean, I could not more agree that also a foundational model is a model for a concrete research question or clinical application question. And in that respect, at least I would say we should be very brilliant and visionary when we talk with our funders, but here we can be more modest and perhaps also a little bit more critical. And I liked very much the discussion. I hope you also enjoyed it. So thank you very much, dear colleagues, for this interesting discussion. Thank you everybody for being here and attending. And now it's lunchtime.